Good morning. So we're going to start Chapter 16, Special Senses, so general and special senses, and we're going to just dig right in. So sensory information is continuously bombarding us, and there are lots of different types of sensations. So we have the basic general sensations like touching, pressure sensors, detecting changes in pressure, receptors that detect hair movement on the arms. We also have taste receptors and hearing receptors. These are special sense receptors. So Chapter 16 is all about the different types of senses that we have. We can classify receptors as very simple. So receptors for touch are considered very simple, as they're just the dendrites reaching out as the receptor that take in information for that bipolar neuron. But they can also be very complex, like your eye. Your eye is a very complex organ of sensory reception that takes in visual information or light rays and interprets them, helps to interpret them as whatever vision you're seeing. Stimuli or sensations, the stimulus that causes the sensation, can be pleasurable, can be painful, can just allow you to detect changes from moment to moment. And some of these stimuli we're not going to be aware of. And this is because not all of the sensations reach our cerebral cortex. Our cerebral cortex is where information that we are consciously aware of is going to be found. Other things are not going to be found there. Other sensations that don't reach the cerebral cortex are not going to, we will not be conscious of. So things like blood pressure changes when you sit and then stand, or the change in your bladder as it starts to fill. You're not actually consciously aware that your bladder is filling. You're only aware when your bladder is full and you have to release. Those are things that we're not going to be aware of. They're not that important. Receptors, to be effective, have to also be able to change information from one form to another form. So the information that comes in could be touch, could be light information, hearing, sound waves, could be chemicals that we taste. They all have to be changed to electrical impulses because our CNS, brain and spinal cord, can only take in electrical impulses, and then they're able to identify, what to do with those, with the information they receive. So to be an effective transducer, you have to have a resting membrane potential, and you have to have either chemically or voltage gated channels. So I don't know if you remember what these look like. This is a little review from last semester, just to help you remember what the plasma membrane of a neuron looks like, and give you a little information about how the action potential occurs as well.
In order to study how action potentials are generated, we will first insert two electrodes into the neuron, one to record the membrane voltage, and another to inject current that can be used to push the membrane voltage toward more positive or depolarizing, or more negative or hyperpolarizing voltages. Let's look at what happens when our model neuron is stimulated. The first two stimuli shown here are insufficient to depolarize the membrane past the threshold level. Thus, no action potential is generated, and the voltage quickly returns to the resting level. However, the final stimulus is large enough to generate an action potential. In this experiment, the current injected into the cell, the stimulus, is represented in the upper graph, and the voltage response below. Notice that a hyperpolarizing current drives the membrane potential in a negative direction, with the voltage response of the cell directly proportional to the magnitude of the injected current. The same relationship is true for small amplitude and duration. The action potential can be
Okay. I'm going to fast forward this because I'm not going to ask you questions about the actual action potential. But I do want you to see the different channels that are associated with the neurons. And so the action potential is what causes the impulse to move from one cell to the next cell. And here you can see the different channels. So remember, you have to have a resting membrane potential, and I usually say resting membrane potential of a neuron is about -70. This is saying -60. And then the threshold mark is about -55. They're saying about -40. So we're a little bit different in what numbers we're using, but the same information is going to apply.
potential. These potassium channels, a subset of all potassium channels, are constantly in the open state. During the rising phase, the increasing positive shift in membrane potential is driven by the opening of progressively more and more voltage-gated sodium channels
We got
and
these
the entry of sodium
voltage-gated
ions into the neuron.
channels.
This inward sodium current depolarizes the membrane voltage, as indicated by the red color shift in the membrane. The membrane potential is now in its most positive state. It has overshot 0 millivolts. At this positive potential, two processes are occurring simultaneously. First, the voltage-gated sodium channels that initially activated during the rising phase begin to close. As a result, sodium conductance starts to decline. Next, potassium channels begin to open, driving the membrane potential back toward the equilibrium potential for potassium. These voltage-gated potassium channels differ from leak potassium channels in that they are normally closed at the resting potential, but open in response to depolarization. Thus the term voltage-gated.
All right, so I'm going to shut this off and go back to the lecture. So our neurons are very effective transducers. They have a resting membrane potential and they have voltage-gated channels that allow them to increase the membrane potential, so cause depolarization to occur and send a signal to the next cell. The receptive field is a region of the body that has receptors on it, and the receptors, the number of receptors in that region will determine the receptive field. So if you have very few receptors, then your receptive field is going to be very large. So they are negatively correlated. The larger the number of receptors you have, the smaller the receptive field you have. So your fingers tend to have a lot of receptors, which means they have a very small receptive field. If you remind me in lab, we're going to actually do a fun activity with the receptive field. And inversely, if you take a different region of the body, like the forearm, you're going to have a much larger receptive field. You don't need as many receptors here. Your fingers are made for taking in sensory information. Your forearms are not. Receptors can also be considered either tonic or phasic. So a tonic receptor is a receptor that continuously sends out stimuli. Receptors have to continuously send sensations so that you recognize, whatever that sensation is. For balance, that would be an example. Constantly sending signals so that you don't lose your balance and become dizzy. Another example of tonic receptors are pain receptors. Pain receptors have to be tonic. And a lot of people think, well, why would you want your pain receptors to be tonic? Because they're constantly sending signals of pain. And that sounds very well, unfun. You know, who wants to continuously be in pain unless you're like some weird masochist, I guess. The reason it's important is because if we don't have that pain, we may forget that we have damage to the body. And so it's really important that we have that constant reminder that we need to get something fixed. Make sense? The other type of receptors are phasic receptors. Phasic receptors phase out. So they stop sending signals. Their sensitivity decreases over time. Things like pressure receptors are phasic. And this is kind of a fun thing to do. You can actually change coins, and of course I have no coins here on my desk now. My desk is usually filled with stuff, but not today. Put coins on your hand. So, whoop, there you go. And you can, eventually, you will no longer feel those coins on your hand. You may know they're there because you know that you put them there, but you will no longer feel them. And so I like to ask this: how many people have ever lost their keys while they were in their hand? Looking for their keys, they're looking everywhere. Aren't they in your hand? Oh, whoops. Because you forget, because the pressure of the keys decreases over time. Most of you are probably sitting or laying down, and you probably forgot that you had pressure on your butt or on your back while you were watching this. But now that I've reminded you, you're probably thinking, oh yeah, there is pressure there. Unless you have damage to those regions, you're not going to be thinking about the pressure. Make sense? So very interesting stuff. And the last thing I'll talk about before I finish this, sorry, I'm going to go back a little. Okay. I want to talk about how we classify receptors. So by where they're located, what their stimulus is, and then the type of stimulating agent. So we have general and special senses. General senses are located all over the body, internally and externally, and they detect basic information like pain, pressure, stretch, temperature, things like that. And then we have special sense receptors that are all located in the head, which we'll talk about towards the end of this chapter. We have the origin of the stimulus can be either external, so from the environment, or internal, like stretch receptors in the stomach when you are eating. That would be an internal stimulus. Proprioceptors are located in the joints, so they detect joint movement. And then the stimulating agent. So stimulating agents can be chemicals, they can be temperature, it can be light, movement, pressure, or pain receptors. Nociceptors are pain receptors. I'm going to stop here. I'll talk about referred pain in my next video. I hope you've enjoyed the first portion of this chapter, and I will talk to you again in a few minutes. So, jump on the next video soon. Bye.